Okay, morning everybody. Thank you for attending this lecture. So today I'm going to tell you about the most important soil-borne diseases of pome fruit, and those actually mostly are on apple. White rot are also on pear, but in general pear trees suffer much less from soil-borne diseases than your apple trees. So we will be looking at 4 diseases. So I think a lot of people underestimate the problem associated with managing soil-borne diseases, and a part of this is because they underestimate that these organisms live in the soil and they really have a function other than infecting plants in itself and using them as a food source. So if the host isn't available, a lot of these pathogens also have what we call a saprophytic phase, so it can live in the soil very happily. So therefore, also when environmental conditions become less conducive, they form these very thick and resistant spores in the soil. So a lot of them stay in the soil for periods from 5 to 10 years without being affected by pretty much anything that we do aside from fumigation. So, and what also contributes to difficulty is that our soil types varies a lot. So therefore there's no one fit solution for managing your soil-borne diseases. So the first disease that we'll talk about is white rot, and that is caused by Rosellinia necatrix. So you can see the characteristic symptoms associated with the disease is this white mycelium that forms on the upper part of the trunk. However, this is not always diagnostic, and especially when you look at the above ground symptoms, this is often cannot be differentiated from Phytophthora. So for all your soil-borne diseases, it's very important to have the identification correctly. So we have a plant disease clinic at Stellenbosch University where you must submit samples to make sure that you have the correct identification of the cause of your soil-borne disease, because as we will go along, you will see that the way that we manage them are different. So very important to not try and guess which soil-borne disease you are working with. Very characteristic, if we make isolations from the disease trees and we look at the pathogen in culture, you see that it forms these very characteristic hyphal pear swellings. But again, this is an identification that must really be done by a specialist, because there are a few closely related fungi that can also form these structures. The problem with managing Rosellinia is that it has a very wide host range, more than 197, and these include a lot of fruit trees, including apples, pears, grapes, citrus. So very difficult once you have it in your orchard to then try and re-establish any other tree crop, because most tree crops are actually susceptible to the disease. It is favored by high organic matter because it is one of those organisms that really can have a phase in the soil without a host plant being present. It has a relatively wide pH there where it can function, and then also a general temperature requirement. The pathogen in itself does not form spores, so these are the reproductive structure of the pathogen. It's very interesting that it doesn't spread via spores, but it actually spreads from root to root contact. So if you have one tree that are infected, if the root of that tree gets to the next tree, then you basically get the disease spreading through the roots. And that is also very important when we come to the management of the disease. And as I previously mentioned, as most of your soil-borne pathogens, it can survive for several years in the soil due to the fact that it can live without a host. As with all plant diseases, you should try and not or avoid stress conditions to your tree, such as over-irrigation or drought stress. However, once you have a disease or an orchard where the trees are starting to show symptoms, you have to basically, those cultural practices will not control your disease, and you have to start removing trees. This is unfortunately the best option currently. So you need to remove the tree that is showing symptoms, as well as 2 trees on the side of each of your symptomatic tree. And you basically need to dig a trench around that tree and make sure that you remove all of the soil. You can either then sieve the soil, or you need to import soil from a different area from your farm, and specifically make sure that if you remove the soil, there's a lot of roots that still contains the pathogen, that you then dispose of it in an area where it will not recontaminate your orchard. So really a very challenging disease to manage, but it is very important that once you see symptomatic trees, that you actually remove them, because otherwise they will, the disease will keep on spreading from tree to tree with root contact, and also with the movement of soil within your orchard. So there are unfortunately no chemical control. Most of the soil-borne diseases, it is a problem because the fungicides that we use are not systemic, so they move very short distances in the soil. So there have been a few in vitro studies where people have looked at several fungicides where fluoxetine has been shown effective. However, as yet, we don't have a way of effectively applying this to soil. So again, back to you have to remove the tree once it is symptomatic. And I think there is also sometimes overestimation of what some of the biocontrol agents can do. Unfortunately, things like trichoderma bacillus, these won't help managing the disease, because the pathogen is already inside of the tree roots. So you cannot try and displace the pathogen by adding biologicals to the symptomatic tree. So the next disease is southern blight. This is caused also by a fungus known as Sclerotium rolfsi. So you can see this is the reason why they call it southern blight, because the whole tree actually dies and dries out. The pathogen, characteristically similar as Rosellinia, will form these white hyphae on the tree, but unlike Rosellinia, it produces a kind of a spore structure which we call sclerotia, which are these, they almost look like brown mustard seeds. And if you look at the life cycle, you have to be even more careful in managing this disease, because unlike Rosellinia, the southern blight pathogen produces spores known as the sclerotia that can be moved with soil. So there are the general conditions that favor the pathogen, almost similar to white rot. Also organic matter, because again it's a pathogen that can survive very well without the host. Managing is very similar to Rosellinia. Once you see a tree that is affected in the orchard, you need to remove it. And also be careful, because in several occasions where we've seen this, the disease actually came from nursery materials. And because, as I already mentioned, it produces these small little resistant spores, it's going to spread faster in your orchard. So immediately once you see the symptoms, make sure that you identify the disease correctly and remove those trees. There is some tolerance in the M9 rootstock, but this will not prevent the tree from actually get diseased. So currently there's no tolerant rootstocks. So the third disease is our Phytophthora diseases. So these can cause stem cankers, as you can see on these photos. And under certain circumstances, it's not very common. We've seen this under net production, where it can actually cause a fruit rot as well. And again, the foliar symptoms are not that characteristic, so make sure to identify or have the disease identified correctly. Then this type of symptom is that our disease clinic identified. It's very interesting, where you can actually, on young trees, also get a blister-like symptom on the above ground cyan, which is unusual, but it can occur. And this is also most likely stressed trees that came from the nursery. The specific Phytophthora species that causes root rot and cankers on apples is known as Phytophthora cactorum. So similar to your white rot, it has a very wide host range, but it will not in general cause problems on pears. This is specifically for apples, and again a pathogen that can survive for very long in the soil. The pathogen is part of a group of fungi or organisms that we refer to as oomycetes, and these are water-loving organisms, so they can actually occur in your irrigation water, so that is the source from where the infection can come from, then also from nursery material, and it can also spread with soil. If we look at the life cycle of the pathogen, again, Phytophthora cactorum forms very resistant structures in the soil, known as oospores, and these are the structures that can live for up to 6 years within the soil or even more. And then, as I already mentioned, it's a water-loving pathogen. So here you can see that the asexual spores actually, under wet conditions, release the swimming spores that can then infect the roots. The zoospores don't swim that far, but it can very rapidly increase in the amount of pathogen in the soil when you over-irrigate. So therefore, as we go to the management, it's very important that the irrigation scheduling is very done appropriately, due to the fact that the pathogen really multiplies under wet conditions. So for the management, similar to the other soil-borne diseases, unfortunately there are no tolerant rootstocks or resistant rootstocks, so there are rootstocks that are less susceptible than others, and we'll get to those specifically. But again, once you have a tree that are stressed, that haven't been irrigated correctly, these tolerant rootstocks give very little control of the disease. It is very important to do proper soil preparation so that you don't get waterlogged soils when you irrigate, and especially do ridging really helps in getting better drainage in the soil to prevent disease buildup. So there is in general the ranking of the different rootstocks. So if you are on a heavy clay soil that you know there's problems with water infiltration or water logging, it's better to choose a more tolerant rootstock. But again, this will not prevent the disease from developing. There are periods of the year when the pathogen is more active, and that is usually during the pink stage in spring. It will start colonizing the host, and then also some of them actually in winter have some activity because they are favored by low temperatures. If we look at chemical control of the disease, once you have an old tree that are already starting to show canker systems, there's unfortunately nothing that one can do to control it. However, when you have young trees, especially young established trees, there are phenylamides that can be used, such as mefenoxam and metalaxyl. So these chemicals can be applied as a soil drench, but only once a year, because they are prone to the pathogen developing resistance. And then also the phosphonates can be used. Unfortunately, currently there's no products registered on apples, so it's difficult to make specific recommendations. Then the last disease is apple replant disease. So this is the stunting of trees when they are replanted onto soil that previously had apple on them, and this is a very complex disease caused by 3 groups of organisms which differ widely in their activity, and this is why basically it's very difficult to control the disease. And this is just some glasshouse results showing that it's not one organism, it's really the interaction of all of these organisms. So if your management strategy isn't going to target all of the organisms, you have a problem and you can't get appropriate disease control. The pathogens